Hello, everyone. I am Alisha Glukhova, and I'm a lab head at VHI, and today I will tell you about single particle electron microscopy. So the outline of my talk is here. So first I will tell you about Cryo as a tool in structural biology, and then I will tell you about how to use single particle Cryo to understand protein structure, and specifically how neutralizing antibodies bind to SARS-CoV-2 spike protein. So I will go over sample preparation and characterization, and then tell you about our standard Cryo processing pipeline, tell you about detectors, movies, classifications, and also how we estimate the resolution, and how we can improve the resolution in the areas of interest. So if we look at the scale of life, structural biology in general and cryo-electron microscopy specifically looks at the most microscopic, at life at the most microscopic level. So it would be right over here. So we use structural biology trying to understand how atoms come together to form structure. In our case, it will be protein structure. So molecular modeling also fits into this category. However, I will not be talking about it today. So there are three main methods that are available to understand protein structure. So it's nuclear magnetic resonance, X-ray crystallography, and cryo-electron microscopy. And here I will tell you a little bit about their drawbacks and their redeeming qualities as well. So nuclear magnetic resonance is a pretty old technique. The pros for this method is that the structure is solved in more native conditions. Its structure also represents an ensemble of conformations. So you can then get information on protein dynamics, and it is a very powerful technique. So you can monitor structural flexibility, conformational exchange rates, allosteric processes, reaction mechanisms. However, it also has quite a few limitations. So first is your proteins will have to be fairly small, so usually less than 40 kilodaltons. It requires a lot of pure labeled protein, can be very expensive. And then because the experiment is done over quite a long period of time, your sample needs to be stable at ambient temperature. And also it's a fairly specialized field and requires a lot of knowledge and expertise. So X-ray crystallography is another technique for structural biology. It used to be one of the most commonly used techniques. There's no limitation on protein size. You can get very high resolution structures, even less than 0.7 angstroms. The method can be relatively fast, so it is suitable for automated data collection, and it can be used for structure-based drug design. However, it also has its own drawbacks. First of all, it requires a lot of protein, usually more than 3 mg, and could be a lot more. And then the main limitation is that in order to do X-ray crystallography, your protein has to form crystals, which is not always the case. So due to flexibility or heterogeneity, this is a very severe limitation. And also X-ray crystallography is prone to what we call crystallization artifacts. So it only gives you a snapshot of a protein in solution, and it can be affected by other molecules in this solution. And then one of the recent techniques, so cryo-electron microscopy, this is the technique I will be telling you about today. So the advantages of this technique is that protein is in more native conditions compared at least to X-ray crystallography. Usually this technique requires less protein. It can also allow to use heterogeneous samples. And recent developments also allow to get dynamic information about your sample. There are, of course, several drawbacks as well. So targets have to be relatively large as compared to the NMR. Usually, the data acquired is at lower resolution than, let's say, X-ray crystallography. However, that limitation is being rapidly overcome, and now there are structures up to 1.2 angstrom resolution for apaferritin. The other potential drawback is that this technique is very computationally expensive, and there's no automation. It takes a lot of time to collect the data and process the data to get the structure you want. So far, it has not been used in a high throughput manner. And high throughput microscopes are expensive. So the overview of how we perform the single particle cryo-electron microscopy experiments is here. So we start with sample preparation. For this, you will need a fairly homogeneous pure sample, then that can be applied to the grid. Then the grid is frozen, put in the electron microscope. Then we collected the data, process the data. So first we go to get the 2D projections, pick particles, align them, and ultimately get to the 3D map. And this is the outcome of this single particle cryo experiment. And then we use this 3D map to build our model, to analyze the data, and to analyze the structure. And so the example of the single particle cryo experiment that I will be talking about today is what we recently done in collaboration with the TAM lab. So we used single particle cryo to understand how neutralizing nanobodies bind to the SARS-CoV-2 spike protein. So living through this pandemic, you'll probably already know that the SARS-CoV-2 is an envelope virus. So it has spike protein on its surface, and it uses those spike protein to interact with the ACE2 receptor that is embedded in a membrane of the cell. So once this interaction is happening, then the virus can enter the cell and start the infection. And in order to prevent this from happening, many labs put a lot of effort into developing antibodies and also nanobodies that can potentially prevent this from happening. So they will block this interaction and infection will not happen. So here is an absolutely amazing result of an MT simulation. So this shows how the spike trimer actually interacts with the ACE2 protein. So here's the spike. It looks a little bit like the ice cream cone. And on its top, there is in green shows the receptor binding domain. So it flips out and interacts with the ACE2 receptor for the target cells. And so because this interaction is required, you can imagine then lots of antibodies and nanobodies target this receptor binding domain specifically to prevent this from happening. And so TAM lab was very much interested in obtaining such neutralizing nanobodies to prevent SARS-CoV-2 from entering the cells. So what they did is they used spike receptor binding domain to immunize llamas. And from those llamas, they screened and identified 50 unique nanobodies. Out of those 50 nanobodies, they realized that they can be grouped into two separate classes, class 1 and class 2. And those two nanobodies from those two classes do not compete with each other, meaning that very likely they bind to two separate epitopes on spike proteins or spike RBD. And what we did is we set out to identify where those epitopes are and hopefully get a structure of spike protein bound to nanobody from each of those classes. So from class 1, we chose nanobody 2, and from class 2, we chose nanobody 10. So the questions that we asked for our structural biology experiment was, first of all, where is the epitope? Can nanobodies bind to the whole spike protein? And how nanobody binding affects the spike conformation? And this was a joint project. We were using both X-ray crystallography and cryo-electron microscopy. So the X-ray crystallography originally was supposed to answer the high resolution information and specifically get the information of what is the epitope. And then we were going to use Cryo to get the structure of nanobodies bound to the whole spike protein to understand what is the spike conformation and can the nanobodies bind to the whole spike. And most of all, we were interested in getting a structure of both nanobodies bound to spike together to get information on the potential for the cocktail therapies where both nanobodies would be used together. So when we do start cryo experiment, the first step is to prepare the sample. And this is very important in structural biology overall, is that your sample has to be good. And that means it has to be pure, homogeneous, and behave fairly well. So be fairly stable and not aggregate. So in this case, Phil from TAM Lab prepared a complex between nanobody 210 and the spike protein. So in that case, the spike was purified from HEK cells and nanobody 2 and 10 from E. coli. So he made the complex by mixing excess of nanobody 2 and 10 and the spike protein. And here you can see the size occlusion profile showing you a single peak for this complex. And this is exactly what we want to see. So homogeneous sample. And if you run that peak on composite gel, again, you can see that this sample is very pure. So this is a band for the spike, and this is the bands for nanobodies, and there's nothing else. So we knew from here that this sample should be very, very suitable for cryo-electron microscopy. However, the next step before cryo is further characterization using the negative stain. And this is very common, so most people do it before they start the actual cryo experiments. And so this method is used to get preliminary structural information on your complex or protein of interest. So for negative stain electron microscopy experiments, what we do is we take our samples, in our case, the spike nanobody complex, we put it on a cryo grid, and then we cover it with the electron dense stain solution. In our case, it was uranyl formate. So something with a very high density. Then we will off the excess stain. And the contrast is built by the leftover stain that's left around the particles. And so what we see is basically the cost on our particles that are surrounded by this excess electron dense stain. So this is how it looks. So our particles, because it's made out of many carbon atoms, it is a lot less electron dense compared to, let's say, uranyl formate. So what we see is the white area where the particle is compared to the very electron dense solution throughout. And so this is the actual micrograph of what we got when we imaged our spike nanobody complex using the negative stain. So you can see darker color micrographs with the white particles throughout. And usually we use this to estimate again how good the sample is. What we're looking for is very little aggregation, which is actually what we see here as well. So the only aggregates are probably here and here. And it is a very homogeneous looking sample as well. Some heterogeneity you see is probably due to different orientations of the particles. So this is the micrograph we saw on the negative stain electron microscopy. What we did is also we analyzed this preliminary to understand what kind of particles we're dealing with. So we picked the particles. So those are just those white particles, white high contrast particles. And then we classify them. So this is shown here. So this is classification of the spike alone, so without bound nanobodies. And you can see this very ice cream shaped particle. So similar to what I showed you before in the MD simulations. If you do the same thing for the complex of spike with, let's say, one of the nanobodies, you can straight away see the difference. So this extra bits, this is the nanobody bound to spike. So even at this stage straight away, you can see whether your, let's say, complexing experiment worked or not. In our case, it did, and we saw a difference when the nanobody was bound. And so, of course, we did this also for the complex of spike with both nanobody 2 and 10, and we deemed that this sample was very good to continue this cryo. So sample preparation for the actual cryo experiment is a bit different than from negative stain. So we start in the same way. So here's we have our grid and the tweezers that hold the grid. We apply our sample in solution in this case. Then we blot off excess liquids, and then we quickly plunge freeze this into, in this case, liquid ethane. So liquid ethane helps to freeze the grid extremely quickly. And so it is needed to form the vitreous ice instead of the crystalline ice, which is what we want for our cryo imaging. And so this is what we expect to see. So this is our grid. In the grids, there it's usually is carbon coated, and then there's these holes where we deposit our protein solution. And after we froze, we hope to see vitreous ice, so in blue here, with the embedded particles. And we hope to see a single area of particles that will be in different orientation. And this is, the whole process is automated nowadays. So we have this machine. So in this case, this is the Vitrobot Mark IV from Thermo Fisher that basically take all of this process and automate it. So this is just a short video to show how it is done with the Vitrobot. So here we apply the sample. There's a little grid over here. Takes about 3 to 4 microliters. Then the grid is blotted with filter paper. So this is this. And after that is done, it will be very quickly plunged. So right here, right now, into the liquidating. So in Melbourne, we have 2 main cryo-electron facilities. So there's Ramacioti Center for Cryo-electron Microscopy at Monash. And then there's the Jan Holmes Imaging Center at Bio21. So their high-end microscopes are all from Thermo Fisher. So the highest end microscope is the Titan, which is a 300 keV microscope. And then the Bio21 facility also has Arctica and Glacius. And the Ramacioti Center has Arctica. The higher the power of the microscope, usually the better resolution you expect. However, both Arctica and Glacius are also capable of producing very high resolution images up to 2 angstroms. They also have different cameras on them, and I will talk about cameras a little bit later. So even though from the outside, the cryo microscope is looking like very much like a box, it is extremely complicated inside. So you can see all these wires, and this is the column for the microscope, and there's a lot going on. However, if we distill this, it is also relatively simple. So all the microscopes, they have the electron gun where the electrons come from at the very top. Then they have a set of lenses and apertures. Then there's a place to put your sample, so the specimen holder, followed by more lenses and more apertures. And then at the end, there's the fluorescent screen or camera. So cryo as a technique has been around for many decades. However, for a while, it was not considered a high resolution technique. And only in the last decade, it became possible to get high resolution structures using single particle cryo. And this is what we call the resolution revolution. And there are a couple of developments that allow for this to happen. So one of them is hardware developments. And one of the most important ones is detectors. So for a while, in the old days, people used film to collect images from electron microscope. However, it was not a very high throughput technique. Then came CCD, so the charge coupled devices. So here, the electrons from the electron beam hit the scintillator, then this information is converted into the photons. And this is transferred to the CCD sensor via the fiber optic cable. However, the problem is that there's a high chance of spreading the signal to the neighboring cells, and readout is fairly slow. However, with the resolution revolution came the direct electron detectors. So they're based on the CMOS sensor, which is the complementary metal oxide semiconductor. And in this case, we don't convert electrons into photons, so they're imaged directly by the sensor. And this reduces the spreading of the charge. And so the advantages of this direct electron detectors, that they are very low noise, They're extremely high resolution and they're fast readout compared to the CCD. And this is what all the high-end microscopes nowadays use. So they all have direct electron detectors. I also should mention that why detectors are so important for Cryo. The reason why the electron detectors are so important for Cryo is because Cryo imaging of biological samples deals with very low signal-to-noise images because biological samples are very prone to radiation damage. So because of those very low signal-to-noise, we do need very good detectors. And in this case, direct electron detectors are the best currently. So how do we quantify how good the detectors are? So for this, we use what's called the DQE, so detector quantum efficiency. So it is a measure of signal to noise that detector can detect at different frequencies. So here's the DQE plots for different detectors. So here on the x-axis is a fraction of the Nyquist frequency. So Nyquist is the smallest possible frequency, distinguishable at given magnification. So Falcon 2, K2 Summit, and D20, they're all direct. I'm not sure about D20, they're direct electron detectors. And you can see they all start fairly high, the DQE, and as soon as we go to higher resolution, it drops. And the higher the number here, the better the detectors are. So the new detectors, there's a Thermo Fisher Falcon 4 and the Gatan K3, supposedly have much higher DQE throughout the whole range. However, the DQE curves have not been published yet, but we've been promised that they're much, much better. And they're certainly faster than the old detectors as well. And so why is the speed of the detector is so important? And the reason behind that is for the fast readouts of detectors allow us to collect movies instead of the still micrographs. So because our signal is low, because of the low signal to noise of biological samples, we need to take long exposures. So usually they last at least a few seconds, sometimes even fractions of a minute. And because of such long exposures, we can observe movements. So it's sample movement. We also get radiation damage from our sample as we accumulate the dose. So the one way to mitigate this, again, is to collect movies instead of stills. So then what we can do, so if we look at a single frame of such movie, the signal to noise is extremely low. So this is an example of such single frame of a movie. You can barely see that there is something there. However, if we average movies during the whole exposure, so for example, this is, I don't know, let's say a couple of seconds of exposures, you can see features of your particles a lot better. However, the problem is that because of this movements like sample movement, for example, those features are blurred. So here's the very blurred particles. However, they're visible very well. And in the old days, when we weren't able to collect the movies, this is what you have. And this severely limited your resolution. However, nowadays, because we collect movies, we can align individual frames and basically correct for the movement. And this is what we get. So not only do we get much higher signal to noise, but we also mitigate and correct the movement as well. And so these aligned movie frames, this is what we work with when we do our Cryo analysis. And there are special programs that do it, for example, the motion correction programs in the Cryo pipeline. So the next step after we got our movie is to estimate what's called a CTF. So CTF is a contrast transfer function, and it describes the signal fallout with increased spatial frequency. So mathematically described how aberrations in a transmission electron microscope modify the image of a sample. So here's the formula, how to identify, to calculate the CTF. And I just want to highlight that it is a function of defocus and spatial frequency. And what we can see from here is that CTF can be modulated with the defocus. And we take advantage of it because during our data collection, we normally collect images at different defocus. So because the biological samples have a very low contrast, so it's both low amplitude and low phase contrast, we use defocus to increase the contrast and to see our particles better. For example, here, I borrowed this image. So this is an image of a virus. So here you can see this same viral particles at the image is taken either at -1 defocus, -1 micron defocus, or at -3. And you can see that here you can see the virus particles a lot better compared to the focus. However, you cannot defocus, and the more defocus you apply, the less high resolution information you can recover. However, we can apply CTF correction to recover at least some high resolution information. But overall, the preference is to minimize defocus as much as possible during the data collection. All right, so how do we estimate CTF from an image? So first, we take our micrographs and then we look at their power spectrum. So power spectrum shows Fourier transform of an image, and it shows how intensity is transferred as a function of the frequency from this image. So you can see those oscillations going. So this is increased frequency, and this the oscillations of the signal. So what we can do is we can fit those oscillations with a function, and then compare how good is our fit to our actual data. So for example, this is done in this picture by the program called CTF Find. So it simulates CTF at different defocus, and then it compares the simulated CTF to the actual image. And then we decide how good is the fit based on the cross correlation between simulation and the actual data. So here's the data and here's the actual fit, and you can see that the fit is relatively good till about here. And this is what we decide as the CTF resolution estimation. So all of that is done to prepare our micrographs for the next steps. So we have our aligned movies, we have our CTF parameters estimated. So from here on, we go on to particle picking. So it's the protocols are similar to what we've done in negative stain. However, the problem here is the low signal to noise of the particles in the micrograph. For example, here is the micrograph of the actual spike nanobody complex, and you can barely distinguish particles here. However, they're there. So for example, if I pick them, so this is the particles that we see. And there are multiple ways to do the particle picking. So there are some different algorithms are based on either just the Gaussian distribution of the density, so that they assume that you have a very dark spots that you're looking for. There are programs that are based on the neural network algorithms, and they're very common to use. It is also very common to use template-based picking. So for the template-based picking, you provide either a 3D volume or 2D projections and ask the program to find those features in these micrographs. These can be very powerful. However, it also has its own potential drawbacks and limitations. So hard to identify particles and need to make sure that they're there. So for the template-based picking, even though it's probably one of the most common ways to pick the particles from micrographs, one needs to be very careful. For example, a while ago, there was a paper where they claimed that they identified new conformation of the HIV gag protein. However, when other people tried to analyze the data, they noticed that there was such a low signal to noise in the micrographs that there were no visible particles. And what it turns out is that because they used template-based picking, they basically picked noise out of noise, then they further aligned to get the structure they wanted from the beginning. So for example, here, this is what's called end-stain from noise. So we can use templates to pick something out of the pure noise. So this is what's shown here. And if you pick sufficient amount of particles from those noisy images, because we use templates to do this, and then if we align sufficient amount of particles, and they can be aligned on each other, and then we recover the similar image to what we started. So there are ways to mitigate this for particle picking, and one of them is to low pass your templates. And this is what's currently done. So again, template picking is still one of the most common techniques for identifying particles in the micrographs. However, it needs to be used with caution. So once we have our particles picked, we extract them, and then we perform 2D classifications. So this is a way to just sort through enormous amount of particles and identify particles that look good and group them together in the classes. So the classes represent the 2D projections of your 3D volume. And so, for example, here, those are the good classes, and the rest is not so good. So the bad classes can arise either from not very good picking. So when we picked, let's say in carbon areas, when we picked area that didn't have any particles in, some particles in here could be broken, and something else could also go on. But in our good classes, what we're looking for usually is the presence of secondary structures, some recognizable features. So in our case, we know that we're looking for something that looks a little bit like an ice cream cone, and that's what we see here. So we select the good classes and then continue to the next step, which would be creating a 3D volume. So we selected our good particles from 2D classifications, and then we try to reconcile all of those particle projections into a single 3D volume. It can be a single 3D volume, it can be multiple 3D volumes. So for example, for initial model and refined 3D, it would be a single 3D volume. When we do 3D classifications, we ask the questions whether we can reconcile all of those particles into multiple different 3D classes. So for example, here, this is the 3D classification of the SARS-CoV-2 spike nanobody complexes. So in that case, I asked the question of whether we can come up with 3 different 3D classes. And this what happened here. And you can straight away see that we have one class that looks very much like what we expect. So it's spike bound to nanobodies, one that looks not so good, but still has the overall shape, and some that looks completely arbitrary. So in this case, I decided that those classes are not good, and this is the class that we should continue further. So if we further use 3D refinement job in Reliant, then we can, in this case, it was, I also used the C3 symmetry. We got to the final structure of the spike bound to nanobodies 2 and nanobodies 10, which you can see here. So straight away from here, you can see that indeed we have our spike proteins are shown in width and then bound to 2 different nanobodies, so 10 and 2. However, the question is, so how good is our structure? So I'm telling you here the resolution is 2.9 angstroms. So how did we determine that? So we used gold standard FSC, or Fourier shell correlation. So this is cross correlation between 2 independent halves of a data set that were refined independently. So during our refined 3D job, we divide all particles into 2 half sets, so half set 1 and half set 2. And they're treated independently throughout different iterations of the refinement. And they're only recombined together for the final reconstruction. So resolution is defined as a frequency in which FSC drops to 0.143. So this is shown here. So this is the FSC curve as a function of the resolution. So there's 0.143, this is 2.9. And that's what we decided it is here. However, even if I tell you that the overall resolution for that structure is 2.9, it does not necessarily mean that it would be 2.9 angstroms throughout the whole structure. And this is where we use the local resolution estimation to understand local differences throughout the structure. So in this case, overall it was 2.9. However, you can see that in the core of the spike protein, the resolution is probably even higher. However, in the areas of interest, which is right here, so our RBD and nanobodies, the resolution is probably not much higher than 7 to 8 angstroms. And so let me give you an overview of the Cryo workflow that we talked about so far. So we started with the raw movies that we motion corrected and aligned together. Then we estimated CTF parameters for those micrographs. Then we picked the particles. In our case, we used the templates. After we picked the particles, we classified them for class 2D. Then we classified them further using a 3D classification and ended up with the refined map. This map can be improved further by using recent developments in our case, it was Reliance software. So those developments are biasing polishing and CTF refinement. And this leads to an increase in resolution of the map. And then from there on, we can ask specialized questions, such as, can we classify our structure further? So can we do heterogeneity analysis? Can we look at protein dynamics? Or can we do any local refinements in the areas of interest? So the software that I had in mind when I was telling you all of this was Reliance. So this is one of the most commonly used software packages. The other one is also Cryo that is also very commonly used. However, there's quite a few softwares that are being developed. So for example, System, Warp, Simple, and I'm sure there are many more that I'm either not familiar with or that are being developed at the moment. So I would like to tell you just a few words about these ways to improve the resolution, so such as the biasing polishing and CTF refinement. So when I talk to you about correcting motion in the beginning, so when we collect our movies and then we use individual frames to align them to each other, this is done using either the whole frames or patches of them. So at that point, we have no idea how our 3D volume looks like. So this is reference-free frame alignment. So in this case, this is a micrograph that was split into 5 by 5 patches, and then each one of those patches was aligned during the motion correction. However, once we get to the high resolution, once we get high resolution information, for example, once we get to the refined 3D stage, we can actually track individual particles in the micrograph. So we go back to the raw movies and then look for our volumes that we reconstructed during our refined 3D run. And then we look at its movement throughout the micrograph. And here you can see that indeed we cannot describe motion just by looking at the patch of a micrograph. So all the particles actually do have independent trajectories. And this is a very powerful technique, and it can usually increase your resolution quite significantly. So another method to enhance the resolution is the CTF refinement. This is also done at the very end, when you already have the information on 3D volume. So again, when we started the data processing, we estimated a single CTF value for the whole micrograph based on tone rings. However, once we have high resolution information, again, we can go back and compare individual particles that we see in those micrographs with our refined volume. And from there on, we can re-estimate the CTF parameters. And you can see here, our estimation actually changed some of them for up to 2 microns, which is actually quite significant. So here, each dot represents a single particle, and the color represents how much its CTF parameter changed compared to the original estimation. So all of those techniques allow us to increase the resolution of the structure overall. However, it does not improve the resolution at the areas of interest, so specifically at the area of RBD and nanobodies. However, what we can do is specifically look at that area. For example, we can take the signal and subtract the signal from the rest of the spike outside of the areas of interest. So what we're left with is RBD bound to nanobody 2 and nanobody 10. And then we can take that and classify it, looking for perhaps spikes that would be bound to all 6 nanobodies at the same time. And this is what we've done here. For example, the yellow is the good class, and you can see that most particles here are intact, so both nanobodies are present. However, among those particles, we also have particles that are incomplete. For example, where nanobody 2 is missing, so for example, here and here. However, this classification works on the assumption that there are spike particles that have all 6 nanobodies bound. So it's basically either all or nothing. And it worked a bit. So we were able to identify a subset of particles that had slightly higher resolution in our areas of interest. For example, we went from 2.9 angstroms overall to 3.8. This is because we have a lot less particles in this reconstruction. However, you can see slight improvements in the definition of the nanobody 2 and nanobody 10 binding site. However, again, while it was sufficient to have some idea of where the nanobodies bind, it would not help us understand any high resolution information, for example, or model the epitopes between the nanobodies and the RBDs. So we went a step further. So instead of looking at all 3 RBDs of the spike at the same time, we decided to focus on just one of them. So again, we subtracted the density for everything outside of the area of interest. So in this case, it was a single RBD bound to nanobody 2 and 10. And then we classify that, in this case, into 6 classes. So straight away, again, you can see that there are classes that have both nanobodies present and some that have none. So presumably what happened is that some nanobody fall off either during sample storage or grid preparation. And so what we have here is a mix of spikes that have nanobodies bound and spikes that do not. And presumably there's also heterogeneity among different RBDs. So even on the same spike, some RBD can have nanobodies bound and some don't. So if you take this, select the good classes, shown here in orange, we can classify them further. But now we will be asking the question of what if individual RBD and nanobodies are flexible in relation to the spike. So this is the classification in these local searches. And you can see that we can actually improve the resolution quite a bit. And among those classifications, we can straight away select a class that has much higher definition of secondary structures for both nanobodies and the RBDs. Again, so we select a good class shown in orange. We can refine it. And here we get to fairly high resolution, actually. So we got to 3.44 overall resolution. And you can see that instead of our original 8 angstroms near nanobodies 2 and nanobody 10, we get somewhere close to 3.8. So this is shown here. And from 3.4 angstrom structure, actually, we were able to build into that and create a model for how our nanobodies 2 and nanobody 10 bound to the RBD. And we use this to answer the actual question. So how do nanobody 2 and nanobody 10 block COVID infection? So if you look at the ACE2 binding site and the receptor binding domain, so you can see, so in purple, so this is the contacts between RBD and ACE2. And if we overlap them with the nanobody and 2 and nanobody 10 binding sites, you can see that there's a considerable overlap for both. So nanobody 2 is more significant than nanobody 10. So what's happening is basically both nanobodies 10 and nanobodies 2, they basically just block ACE2 from interaction with the RBD. Because we have fairly high resolution structure, we were also able to identify specific contacts between RBD and the CDR of the nanobodies. So this is shown for nanobody 10. And this is also for nanobody 2. I should also say that Phil also had a crystal structure of nanobody 2 bound to the RBD. And these contacts are actually taken from the crystal structure. However, they overlap almost identically with the Cryo structure. So the take-home messages from today's talk are that first of all, for a chance at high resolution structure, you need good sample. You need to be vigilant to produce real structure and make sure that you would not introduce template bias at any step of the processing. The overall resolution of the structure is not necessarily the same as local resolution. So even though your overall resolution is high, there are some areas of interest that can be fairly low. Cryo is a very powerful technique and has the ability to process heterogeneous samples. I was not talking about this, but Cryo is also quite powerful in getting the dynamic information. For example, if you have multiple different conformations of your complexes and proteins, and then you can separate them. And there are also, it's a fairly new technique, so there are constant improvements in both hardware and software, and they try to push the limits. And you just need to constantly stay updated with what's going on in the field. And with that, I'd like to acknowledge the TAM Lab, so Wei-Hon and Phil, and other TAM Lab members. So of course, this work would not be possible without any of them. So from my own lab, I would like to acknowledge Katrina, then Structural Biology Division, Andrew for his help with imaging, and also Ramotiot Center for Cryo Microscopy, as well as the Jan Holmes Imaging Center. I would like to acknowledge Australian government for funding, also VHI and Massive and NCMAS for providing computational resources, and I'm funded by CSL Centenary Fellowship. And thank you all for listening. I am looking forward to taking your questions on Monday. Thank you.